Hi everyone, it's me Akila and welcome to my slide. Today we are going to learn about electron transport chain. Electron transport chain is defined as an electron transferred by NADH and FADH2 through four major protein series to combine with oxygen producing water. Electron transport chain occurs in the inner membrane of mitochondria. The inner membrane is a fluid-filled space between layers. It consists of four major integral protein complexes. These are the four integral protein complexes that act as a carrier protein as well as a proton pump. Protein complex 1 is a large enzyme complex. Next, protein complex 2 is the only membrane-bound enzyme in the citric acid cycle. Then, protein complex 3 has a beautiful dimeric structure. Last but not least, protein complex 4 is a large enzyme of the inner mitochondrial membrane. Two small electron mobile carriers are linked with each protein, which is ubiquinone Q and cytochrome C. Ubiquinone Q, also known as coenzyme Q, is an electron carrier only, not a proton pump. It is a part of the pathway for synthesis of ATP. Also, to carry electron from one complex of integral protein to another. Cytochrome C has a primary structure that consists of about 100 amino acids. It functionally involved in the electron transport chain of mitochondria as an electron carrier from one complex to another. There are two shuttles that occur, which is malonyl aspartate shuttle and glycerol phosphate shuttle. Malonyl aspartate shuttle allows the electron to move across the impermeable membrane between the cytosol and the mitochondrial matrix. The electrons are created during glycolysis and are needed for oxidative phosphorylation. Glycerol phosphate shuttle allow the NADH synthesized in the cytosol by glycolysis to contribute to the oxidative phosphorylation pathway in the mitochondria to generate ATP. It can be found in fungi, animals, and plants. First, NADH donates electron to protein complex 1. 2. Protein complex 1 transfers the electron to ubiquinone Q. 3. Electron from Q moves to complex 3 and transferred to cytochrome C. 4. Cytochrome C carry the electron to complex 4. Then, the electron unites with H+ and O2 in the matrix to form H2O. 1. FADH2 donates electron to complex 2. 2. Protein complex 2 transfers electron to ubiquinone Q. 3. Q carry the electron to complex 3 and transfer to cytochrome C. Cytochrome C carry the electron to complex 4, then the electron unites with H+ and O2 in the matrix to form H2O. In conclusion, the protein and electron carriers alternate between redox reaction. Second, gradient potential of electron and proton can increase. Third, FADH2 produces less number of protons compared to NADH, but this movement will contribute to chemiosmosis.